So, moving ahead, moving ahead with this topic, that is the type 3 restriction enzyme. These restriction enzymes are not used for gene cloning. Okay, unlike the type 2 where it is used for gene cloning, the type 3 are not used for gene cloning. They are the intermediate enzyme between type 1 and type 2 restriction endonuclease. They requires magnesium ion just like type 1 and type 2 and ATP for cleavage just like type 1 requires ATP. So if you remember the comparative difference, type 1 and type 3 will require magnesium as well as ATP, whereas type 2 requires only magnesium. And they cleave the DNA at a well-defined site in the immediate vicinity of the recognition site. If you remember that type 1, they identify a recognition site or a target sequence, but basically they are going to cut 1,000 base pair away. Okay. Whereas the type 2 restriction enzyme, wherever they identify the restriction enzyme site or the target site, they're exactly going to cut at that specific position. Whereas the type 3 are going to cut very close, say for example 26 base pair away, not very far or not exactly at the same site. I hope you get a difference between the three of them. The example of a type 3 is HINF3 enzyme. Okay. Now, how are we going to name these restriction enzyme or restriction endonucleases? How will, we have been saying eco R1, eco R5, SMA1, SMA5, and so many enzymes we have been naming. So, but how basically are we going to name these restriction enzyme? So here let us take the example of HIN3. Now, the first letter of the genus name H, if you look at this letter H here, H stands for Haemophilus, and the first two letters of the species name influenza. So HIN stand for the capital H stands for the genus, the small 2IN stands for the species. Now, what is this D? This D is the strain type. As you know, in bacterial, you will have many other strain. Okay, so HIND, when I say HIND, D represent the strain type of the bacteria. And 3 is for the third enzyme discovered in that organism. So from the same organism itself or from the same bacteria itself, you can get many restriction enzyme, and based on which was the first one to be discovered, which was the second, third, fourth, fifth, so on and so forth, this last numbering, a Roman numbering, is given to them based on that. If and HIN3 being the third one to be discovered, it is named accordingly, that is HIN3. I hope it is very clear how we give the names to these restriction endonucleases. So these are some of the example like BAMH1, the organism from which it is derived. It is Bacillus amylo liquefacient. Okay. So BAM, B stands for the name of the genus, AM stands for the name of the species, A stands for the name of the strain, and 1 stands for the first to be discovered from this organism. So on and so forth. If you look at this table, you can find that how the name has been given to them. In the third column, you can see that there is also a target sequence or a cut at which will take place between these will be the recognition site, and not only the recognition site, but where the star mark that is highlighted, that star mark is where the restriction enzyme is going to cut. Okay. So just a repetitive again, the nomenclature of restriction enzyme consists of 3 parts. It indicates the genus and species of specific organism by using abbreviation into 3 letters. Number 2, it indicates the strain of the relevant species by using a letter, number, or its combination. And third, it indicates specific restriction modification system present in the same organism or strain by using a Roman numeral. Okay. I will not repeat. I know that example of a HIN3 has made it very, very clear. Okay. So example, again, example of the EcoR1 restriction enzyme, just to make it even more clearer. If you see EcoR1, E stands for Escherichia, the name of the genus. CO. coli stands for the name of a species. R stands for the strain, that is RY13. And 1 is the first identified in the bacteria. Okay. Now, I hope it is very clear whenever we talk of restriction enzyme, why they have such particular name, because sometimes we'll feel it's such a weird name, we call them EcoR1, HIN3, okay? But now I think it is very clear why these restriction enzyme do have such specific nomenclature. Okay, even here, these are some of the name of the microorganisms, some of the restriction enzyme, the cleave site, and the cleavage being products. Okay, the cleavage products that are being produced by these restriction enzyme. Okay. Now, we have seen that there are 3 types of restriction endonucleases, right? Those are endonucleases. But here, we will see we also have enzymes that are exonucleases. Endo, the word endo itself means within, okay? They are going to cut the DNA within the DNA strand. But exo, they are going to cut at the terminal or at the end of the DNA. So that is the difference between endo and exo. So the exonuclease is an enzyme that removes nucleotides from the end of the nucleic acid molecule. And an exonuclease would remove nucleotide from the 5' or the 3' end. So basically these exonucleases, not necessary that they are going to cut only from the 5' end, but they can cut from the 3' end also. So, and exonuclease never produces internal cut. Just like how we have seen in the type 1, type 2, and type 3 restriction endonucleases, where if they recognizes a target sequence, they are going to cut the specific DNA sequences, and they are also going to produce the cut, okay, the fragments. So it means that they are going to cut the DNA in between. Whereas these exonucleases, because they are not targeting the internal or the endo part of the DNA, they are only going to cut or chop or cleave the fragment, the DNA from the terminal. Okay. I think it is very clear if you look at this example, the exonuclease activity, how they are going to remove the terminal part of the DNA. So in recombinant DNA technology, various type of exonuclease are employed, like exonuclease BAL31, E. coli exonuclease 3, and lambda exonuclease. So these are some of the examples. Let me just correct this. It should be in italic. Okay. So I hope it is very clear. So we need both exo and endonucleases in recombinant DNA technology, not only cutting from between, but also cutting from the terminal. Now, we also have restriction enzymes, like, we give them a different name sometimes. So we have here, number 1, isosizomers, we call them. Now, what is isosizomers? They are pair of restriction enzyme that recognizes the same recognition sequence. So at the beginning of the slide, I've told you that every restriction enzyme have their own recognition site, okay? But sometimes there are some restriction enzymes that share the target sequence, which means they have the same exact target sequence. The cut site may be same or may be different, but yes, most of the time it is going to be the same. That is why we have the word iso, which means same, okay? And they cut also at the same location. Best example here we have seen SPH1 and BBU1. So these two restriction enzymes are isosizomers. Why? Because if you look at the sequence, we have CGTACG, but the cut is between C and G. Clear? So such enzyme also do occur or do are present, where they are going to cut, identify the same recognition site, and also cut at the same site. For such kind of restriction endonucleases or restriction enzyme, we call them isosizomers. Okay. Now we also have neosizomers. Now what are neosizomers? They recognize the same sequence but cut it differently. Okay, the sequence is same, but the cutting of the enzyme or the location or the site is different here. SMA1 recognizes GGGCCC. Example, XMA1 also recognizes GGGCCC. But SMA1 will produce blunt end because it is going to cut exactly in between the recognition site, that is GGG, and there will be a cut. So there is basically a cut between G and C. Whereas in XMA1, the cut is between guanine and guanine, okay, producing overhangs. The last type of enzyme are also isoquadromers. What is isoquadromers? They recognizes slightly different sequence but produce the same end. So we will see with some example. Example, we have SAU3A and BAMH1. Both give GATC sticky ends. Okay, this is the best example. If you look at this figure, you will see that N represent, or N stands for any nucleotide. So how BAMH1, BGL2 and SAU3A have the same, the first two nucleotide, if you look in BAMH1, the first two nucleotide are different, but then we have CCTAG. Similarly for BGL2, we have NN, we have TCTAG, and SAW3A, we have NNNCTAG. But if you look at the overhangs, that is basically what isoquadromers are all about. These isoquadromers have the same overhang. So sometimes what happens when we cut the DNA sequence with these enzymes, I cut DNA1 with BAMH1 and I cut DNA2 with SAU3A. The overhangs, there is high chance that they are going to ligate. Why? Because these overhangs will be complementary, even if they are being cut by BAMH1 or SAU3A. So such enzymes are also known as isoquadromers. Okay. So in short, I have prepared this table where you have a comparative properties of restriction enzyme. So here you can see the type 1, the type 2 and the type 3. So go through the slide, just try to minimize so that at one look you can look or you can see the major difference between the three type of restriction endonucleases. Okay. Okay, so next moving on to the frequency of recognition sequences in a DNA molecule. Now, what is the frequency that a recognition sequence will be produced in a molecule? How many times? Like if I'm cutting a DNA molecule by a particular enzyme, say EcoR1 or BAMH1 or HIN3, how many times or how many fragments it is going to produce? Okay, or where will these sequences be present? How many times? Okay, the frequency, like how many times. So the number of recognition sequences for a particular restriction endonuclease in a DNA molecule of known length can be calculated mathematically. First, if it is a tetranucleotide sequence, so for example, if a restriction enzyme identify a tetranucleotide sequence, like for example here we have GATC. So how GATC, how many times will it occur? It should occur every 4 to the power 4 times. That is 256 nucleotide. So if you have GATC here, then after 256 nucleotide, you are going to again find that sequence GATC. Then again 256 nucleotide away, you are going to again find the sequence GATC. Okay, so the probability is 4 to the power 4. 4 because you have 4 nucleotide. Okay, 4 nucleotide, and this 4, another one 4 represent the number of because it is a tetramer. Okay, so that is why you have 4 representing the 4 different 4 nucleotide, the ATGC, and to the power 4 representing the nucleotide being a tetranucleotide. Okay. Similarly, if it is a hexamer, nucleotide, you will have six nucleotides, say for example GGATCC, and once every 4 to the power 6, again 4 representing ATGC and 6 representing the hexamer nucleotide sequence. And after every 4096 nucleotide, it will occur again. So if you find GGATCC here in position number 1, then after 4096 nucleotide, you are going to find GGATTC again. Okay. I hope it is clear the frequency at which they will keep on repeating in the DNA molecule. Now, if you're going to perform this in the lab, okay, if you have isolated the DNA, say for example of a plant, and you want to cut the genome of that DNA, of that plant DNA, by any restriction enzyme. So what is the standard setup? So if you look here, the DNA have to be of 1 microgram, okay? Yeah. And then the restriction enzyme, depending on what enzyme, from where have we ordered. So basically, 20,000 units per ml in 50% glycerol. This, of course, comes synthesized by every company. Different company have their own buffers, and all these calculation part or the unit, how we order also will matter. So the restriction enzyme, we will require 0.5 to 10 micro unit per reaction. So for every reaction, the reaction can be 0.5 to 1, the volume by which we are going to add. Okay. Now the buffer have to be compatible, mostly from wherever we are ordering these restriction enzyme, those company will synthesize the buffer also for that enzyme. Okay. And there we have to add 1x. And BSA also we have to add. The BSA that we are going to order will come in 100x, but the working stock will be 1x. The sterile water will make up to the volume as per needed, and the total volume should be 50 microliter. And the reaction, once you have added all these components together, the reactions are incubated for 12 to 18 hours at 37 degrees centigrade. And after that, once you are done with this, you will run it in the gel electrophoresis, and basically this is how you're going to see the bands. Okay, so if you look at this agarose gel, you will see the electrophoresis analysis of restriction enzyme digested for this DNA, FEGABA01 DNA. Okay, FEG ABA01 genomic DNA digested with EcoR1. So we have the same DNA digested by different enzyme or different RE. The second lane represent, the first one represent the DNA itself, okay? The DNA of this FEGABA01. The second lane will represent the DNA that is digested by EcoR1. The third lane will be represented by HIN2, HIN2. The fourth will be represented by NSIL. The fifth will be presented by PSD1. The sixth will be represented by SAL1, SAL1, and lane 7 by SCE1, lane 8 by SEXA1, and lane 9 by SPE1. Okay, so basically the same DNA will be digested by different restriction enzyme, and this is how the band will be created, or this is how the band will be seen. Okay, will be fragmented. So the DNA basically will produce a different profile if we use different restriction enzyme. Okay. So this is the example of the restriction enzyme at the types and classification. So you can just go through this table. I'll not say much because it's already been spoken or taught, but just go through. This is just like a summary. Now the application, In various applications related to genetic engineering, DNA is cleaved by using these restriction enzymes. So if I want to cut the DNA of plant A and plant B, so I can use these restriction enzymes, which are the molecular scissor. Now, they are used in the process of insertion of genes into the plasmid vector during gene cloning and protein expression experiment. So, when I want to incorporate my gene of interest into plant A, say for example, I'm taking a gene from plant B and want to insert it in plant A. So with the help of these restriction enzyme also, I'm going to cut from both the DNA. For plant A, I'm going to cut open the genome. From plant B, I'm going to cut and take out the gene of interest and insert it into the plant A, and to distinguish gene alleles by specifically recognizing single base changes in DNA known as single nucleotide polymorphism. So if there is any difference at a specific nucleotide also, we can use the application of these restriction endonuclease to identify those SNPs, we call them. Okay, so my dear student, I hope you have understood the topic restriction endonucleases. But to know it more further, especially if you want to prepare it for some competitive exam like NET and all, we'll be doing some of the numerical in the next class. So thank you so much. And if you have any queries, we will discuss together in the next class. Thank you.